Hello friends, welcome to Medical Devo. In this video, we will discuss the chapter 19 of 11th Ncert Biology, Excretory products and their elimination. So all NEET aspirant must watch this video. In this video, we will read the chapter and discuss the important point regarding exam point of view. So without further delay, let us discuss this chapter, Excretory products and their elimination. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, chlorine, phosphate, sulfate, etc., either by metabolic activities or by other means like excessive ingestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of this substance with special emphasis on common nitrogenous waste. Ammonia, urea and uric acid are the major form of nitrogenous waste produced by the animals. Here you have to remember that ammonia, urea and uric acid are major forms of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with a minimum loss of water. Let us see the process of excreting ammonia is ammonotelicism. Many bony fishes, aquatic amphibians, and aquatic insects are ammonotelic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surface or through gill surface in fishes as ammonium ion. Kidney do not play any significant role in its removal. Terrestrial adaptation necessitates the production of lesser toxic nitrogenous wastes like urea and uric acid for conservation of water. Mammals, many terrestrial amphibians and marine fishes mainly excrete urea and are called ureotelic. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidney. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, land snails, and insects excrete nitrogenous waste as uric acid in the form of pellet or paste with a minimum loss of water and are called uricotelic animals. A survey of animal kingdom presents a variety of excretory structures. In most of the invertebrates, these structures are simply tubular forms, whereas vertebrates have a complex tubular organs called kidney. Some of these structures are mentioned here. Protonephridia or flame cells are the excretory structures in Platyhelminthes, that is flatworm. Example is Planaria. Rotifers, some annelids, and the cephalochordate Amphioxus. Protonephridia are primarily concerned with ionic and fluid volume regulation, that is osmoregulation. Nephridia are the tubular excretory structure of earthworm and other annelids. Nephridia helps to remove nitrogenous waste and maintain a fluid and ionic balance. Malfingial tubules are the excretory structure of most of the insects, including cockroaches. Malfingial tubules helps in the removal of nitrogenous waste and osmoregulation. Antennal glands or green glands perform the excretory function in crustaceans like prawns. Let us see the human excretory system. In human, the excretory system consists of a pair of kidney, one pair of ureters, a urinary bladder, and a urethra. Figure 19.1. Kidney are reddish-brown, bean-shaped structures situated between the level of last thoracic and third lumbar. Kidneys are situated between the last thoracic and third lumbar vertebra close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in width, and 2 to 3 cm in thickness, with an average weight of 120 to 170 grams. Towards the center of the inner concave surface of the kidney is a notch called a hilum, through which ureter, blood vessels, and nerves enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis with projection called a calces. The outer layer of kidney is a tough capsule. Inside the kidney there are two zones, an outer cortex and an inner medulla. The medulla is divided into few conical masses, medullary pyramid, projecting into calyces, singular calyx. The cortex extends in between the medullary pyramids as renal columns, called a column of Pct. Here you have human urinary system. Here they mentioned the inferior vena cava, adrenal gland above the kidney, renal artery with red color and renal vein with blue color, kidney, dorsal aorta, ureter, urinary bladder, urethra. Inside the kidney there is cortex region, then medulla in between the medullary pyramids, pelvic region, inferior vena cava. Let us see the human excretory system. We saw some points. That each kidney measures 10 to 12 cm in length, 5 to 7 cm in width, and 2 to 3 cm in thickness, with an average weight of 120 to 170 gram. Toward the center of inner concave surface of the kidney is a notch called a hilum, through which ureter, blood vessels, and nerve enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis, with projection called a calyces. The outer layer of the kidney is tough, called a capsule. Inside the kidney, there are two zones, an outer cortex and an inner medulla. The medulla is divided into a few conical masses, medullary pyramids, projecting into the calyces. The cortex extends in between the medullary pyramids as renal columns called a column of Pct. Figure 19.2 represents the column of Pct. Here they mention the longitudinal section diagrammatic of kidney. Here is the calyx, renal artery, renal vein, renal pelvis, ureter, medullary pyramids, renal columns, cortex, renal capsule. Each kidney has nearly 1 million complex tubular structures called nephrons. Figure 19.3 is a figure of nephron, which are the functional units. Nephrons are the functional unit of kidney. Each nephron has two parts, the glomerulus and the renal tubules. Glomerulus is a tuft of capillaries formed by the afferent arterioles, a fine branch of renal artery. Blood from the glomerulus is carried away by an afferent arterioles. The renal tubules begin with a double-walled cup-like structure called a Bowman's capsule, which encloses the glomerulus. Glomerulus, along with Bowman's capsule, is called the Malpighian body, as renal corpuscle. In figure 19.4 represent. The tubule continues further to form a highly coiled network called proximal convoluted tubule (PCT). Diagrammatic representation of a nephron showing blood vessels, duct, and tubules. Here they have mentioned the afferent arterioles, then glomerulus, Bowman's capsule, PCT, DCT, collecting duct, vasa recta, ascending limb of loop of Henle, descending limb of loop of Henle. A hairpin-shaped Henle's loop is the next part of the tubules, which has a descending and an ascending limb. The ascending limb continues as another highly coiled tubular region called a distal convoluted tubule (DCT). The DCTs of many nephrons opens into a straight tube called a collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the calyces. The Malfingiae corpuscle, Pct and Dct of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. In majority of nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. In some of the nephrons, the loop of the Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephrons. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubules, called the peritubular capillaries. A minute vessels of this network run parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephrons. Let us see the figure of Malfingiae body, also called as renal corpuscle. Here they represent a afferent arteriole, and Bowman's capsule. Here is the PCT, proximal convoluted tubule. Let us see the process of urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion, that takes place in different parts of the nephron. The first step in urine formation is the filtration of blood, which is carried out by the glomerulus and is called a glomerulus filtration. On an average, 1100 to 1200 ml of blood is filtered by the kidney per minute, which constitutes roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers, that is, the endothelium of glomerulus blood vessels, the epithelium of Bowman's capsule, and a basement membrane between these two layers. The epithelial cells of Bowman's capsule, called podocyte, are arranged in an intricate manner so as to leave some minute space called a filtration slit or slit pore. Blood is filtered so finely through these membranes that almost all the constituent of the plasma except the protein pass on to the lumen of the Bowman's capsule. Therefore, it is considered as a process of ultrafiltration. The amount of filtrate formed by the kidney per minute is called glomerular filtration rate, or GFR. GFR in a healthy individual is approximately 125 ml per minute, that is 180 liters per day. The kidney has built-in mechanism for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus, JGA. JGA is a special sensitive region formed by cellular modification in the distal convoluted tubule and afferent arterioles at the location of their contact. A fall in GFR can activate the JG cell to release renin, which can stimulate the glomerular blood flow and thereby the glomerular filtration rate back to normal. A comparison of the volume of the filtrate formed per day, 180 liters per day, with that the urine released, 1.5 liters, suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubules. This process is called a reabsorption. The tubular epithelial cells in different segments of nephron perform this either by active or passive mechanism. For example, substances like glucose, amino acid, sodium, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous wastes are absorbed by passive transport. Reabsorption of water also occurs passively in the initial segment of the nephron. Figure 19.5 represents this. During urine formation, the tubular cells secrete substances like H+, K+, and ammonia into the filtrate. Tubular secretion is also an important step in urine formation, as it helps in maintenance of ionic and acid-base balance of body fluids. Let us see the function of the tubules. PCT. PCT is lined by simple cuboidal brush border epithelial, which increase the surface area for reabsorption. Nearly all the essential nutrients and 70 to 80% of electrolyte and water are reabsorbed by this segment. PCT also helps to maintain the pH and ionic balance of the body fluids by selective secretion of hydrogen ion, ammonia and potassium ion into the filtrate and by absorption of bicarbonate from it. Henle's loop. Reabsorption is minimum in its ascending limb. However, this region plays a significant role in the maintenance of high osmolarity of medullary interstitial fluid. The descending limb of loop of Henle is permeable to water but almost impermeable to electrolyte. This concentrates the filtrate as it moves down. The ascending limb is impermeable to water but allows transport of electrolyte actively or passively. Therefore, as the concentrated filtrate passes upward, it gets diluted due to the passage of electrolyte to the medullary fluid. Let us see the distal convoluted tubules function. Conditional reabsorption of sodium and water takes place in this segment. DCT is also capable for reabsorption of bicarbonate and selective secretion of hydrogen ion, potassium ion and ammonia to maintain the pH and sodium-potassium balance in blood. Collecting duct. This long duct extends from the cortex of the kidney to the inner part of the medulla. Large amount of water could be reabsorbed from this region to produce a concentrated urine. This segment allows passage of small amount of urea into the medullary interstitium to keep up the osmolarity. It also plays a role in maintenance of pH and ionic balance of blood by the selective secretion of H plus and K plus ion. Let us see the figure 19.5. Reabsorption and secretion of major substances at different parts of the nephron. Arrow indicate direction of movement of material. Here is the PCT and here is the DCT. Upper region is cortex and inner region is medulla. In cortex region, in PCT, there is the exchange of bicarbonate, NaCl, water, nutrient K plus and H plus ammonia. And in DCT, there is NaCl, water, bicarbonate, K plus and H plus. Descending limb of loop of Henle is permeable to water, so it concentrates the urine, while the ascending limb of loop of Henle is impermeable to water, so it dilutes the urine. It exchanges NaCl. In collecting duct, some amount of water and urea are released into the interstitial space. Let us see the mechanism of concentration of the filtrate. Mammals have the ability to produce a concentrated urine. The Henle's loop and the vasa recta play a significant role in this. The flow of filtrate in the two limbs of Henle's loop is in opposite direction and thus forms a counter current. The flow of blood through the two limbs of vasa recta is also in a counter current pattern. The proximity between the Henle's loop and vasa recta as well as the counter current in them help in maintaining and increasing osmolarity towards the inner medullary interstitium, that is from 300 ml osmol per liter in the cortex to about 1200 osmol per liter in the inner medulla. This gradient is mainly caused by NaCl and urea. Here you have to remember that the gradient is caused due to NaCl and urea. NaCl is transported by the ascending limb of Henle's loop, which is exchanged with the descending limb of vasa recta. NaCl is returned to the interstitium by the ascending portion of vasa recta. Similarly, small amount of urea enters the thin segment of the ascending limb of Henle's loop, which is transported back to the interstitium by the collecting tubules. The above described transport of substance facilitated by a special arrangement of Henle's loop and vasa recta is called the counter current mechanism. Figure 19.6. This mechanism helps to maintain a concentration gradient. Diagrammatic representation of a nephron and vasa recta showing countercurrent mechanism. Here, the concentration is 300 osmol per liter. Then it goes on increasing to 1200 ml. Here they mentioned the afferent arteriole, efferent arteriole, Bowman's capsule, glomerulus, exchanging material as we explained earlier. In the medullary interstitium, presence of such interstitial gradient help in an easy passage of water from the collective tubules, thereby concentrating the filtrate urine. Human kidney can produce urine nearly 4 times concentrated than the initial filtrate form. Let us see the regulation of kidney function. The functioning of the kidney is effectively monitored and regulated by hormonal feedback mechanism involving hypothalamus, JGA, to a certain extent the heart. Osmoreceptors in the body are activated by changes in blood volume, body fluid volume, and ionic concentration. An excessive loss of fluid from the body can activate this receptor, which stimulates the hypothalamus to release antidiuretic hormone ADH or vasopressin from the neurohypophysis. ADH facilitates water reabsorption from the later part of the tubule, thereby preventing diuresis. An increase in the body fluid volume can switch off the osmoreceptor and suppress the ADH release to complete the feedback. ADH can also affect the kidney function by its constrictory effect on the blood vessels. This causes an increase in blood pressure. An increase in blood pressure can increase the glomerular blood flow and thereby the glomerular filtrate rate. The JGA, juxtaglomerular apparatus, play a complex regulatory role. A fall in glomerular blood flow per glomerular blood pressure (GFR) can activate the JG cell to release renin, which converts angiotensin in blood to angiotensin I and further to angiotensin II. Angiotensin II, being a powerful vasoconstrictor, increases the glomerular blood pressure and thereby GFR. Angiotensin II also activates the adrenal cortex to release aldosterone. Aldosterone causes reabsorption of Na plus and water from the distal part of the tubules. This also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as renin-angiotensin mechanism. An increase in blood flow to the arteria of the heart can cause the release of atrial natriuretic factor (ANF). ANF can cause vasodilation, dilation of blood vessel, and thereby decrease the blood pressure. ANF mechanism therefore act as a check on the renin-angiotensin mechanism. Let us see the maturation. Urine formed by the nephrons is ultimately carried to the urinary bladder, where it is stored till a voluntary signal is given by the central nervous system. This signal is initiated by stretching of the urinary bladder as it gets filled with urine. In response, the stretch receptor of the walls of the bladder sends signal to the CNS. The CNS passes on motor messages to initiate the contraction of smooth muscle of the bladder and simultaneous relaxation of the urethral sphincter, causing the release of urine. The process of release of urine is called a maturation, and the neural mechanism causing it is called a maturation reflex. An adult human excretes on an average 1 to 1.5 liters of urine per day. The urine formed is a light yellow colored watery fluid which is slightly acidic, pH is 6, and has a characteristic odor. On an average, 25 to 30 grams of urine is excreted out per day. Various conditions can affect the characteristics of urine. Analysis of urine helps in clinical diagnosis of many metabolic disorders, as well as malfunctioning of the kidney. For example, presence of glucosuria, and ketone bodies, ketonuria in urine are indicative of diabetes mellitus. Let us see the role of other organs in excretion. Other than the kidney, lungs, liver, and skin also help in elimination of the excretory waste. Our lung remove large amount of CO2, approximately 200 ml per minute, and also significant quantities of water every day. Liver, the largest gland in our body, secretes bile-containing substances like bilirubin, biliverdin, cholesterol, degraded steroid hormone, vitamins, and drugs. Most of these substances ultimately pass out along with digestive wastes. The sweat and sebaceous gland in the skin can eliminate certain substances through their secretion. Sweat produced by the sweat glands is a watery fluid containing NaCl, small amount of urea, lactic acid, etc. Though the primary function of sweat is to facilitate a cooling effect on the body surface, it also helps in the removal of some of the waste mentioned above. Sebaceous glands eliminate certain substances like sterol, hydrocarbon, waxes through sebum. This secretion provides a protective oily covering for the skin. Do you know that small amount of nitrogenous waste could be eliminated through saliva too? Yes. Disorders of the excretory system. Malfunctioning of the kidney can lead to accumulation of urea in blood, a condition called uremia, which is highly harmful and may lead to kidney failure. In such patients, urea can be removed by a process called hemodialysis. During the process of hemodialysis, the blood drained from a convenient artery is pumped into a dialyzing unit called artificial kidney. Blood drained from a convenient artery is pumped into a dialyzing unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tube surrounded by a dialysing fluid having the same composition as that of plasma, except the nitrogenous waste. The porous cellophane membrane of the tube allows the passage of molecules based on concentration gradient. As nitrogenous waste are absent in the dialysing fluid, these substances freely move out, thereby clearing the blood. The cleared blood is pumped back to the blood through a vein after adding antiheparin to it. This method is known for the thousands of uremic patients all over the world. Kidney transplantation is the ultimate method in the correction of acute renal failures, kidney failure. A functioning kidney is used in transplantation from a donor, preferably a close relative, to minimize its chances of rejection by the immune system of the host. Modern clinical procedures have increased the success rate of such a complicated technique. Renal calculi, stone or insoluble mass of crystallized salts, oxalate, etc. formed within the kidney. Glomerular nephritis, inflammation of glomeruli of kidney. Let us see the last part of this chapter summary. Many nitrogen containing substances like iron, CO2, water, etc. that accumulate in the body have to be eliminated. Nature of nitrogenous waste formed and their excretion vary among animals, mainly depending on the habitat. Availability of water, ammonia, urea, and uric acid are the major nitrogenous waste excreted. Protonephridia, nephridia, Malpighiae tubules, green glands, and the kidney are the common excretory organs in animals. They not only eliminate nitrogenous waste but also help in maintenance of ionic and acid-base balance of the body fluids. In humans, the excretory system consists of one pair of kidney, a pair of ureter, a urinary bladder, and a urethra. Each kidney has over a million tubular structure called nephrons. Nephrons is functional unit of kidney and has two portions, glomerulus and renal tubules. Glomerulus is a tuft of capillaries formed from afferent arterioles, fine bunches of renal artery. The renal tubule starts with a double-walled Bowman's capsule and is further differentiated into the proximal convoluted tubule (PCT), endless loop, and distal convoluted tubule. The TCTs of many nephrons join to a common collecting duct, many of which ultimately open into the renal pelvis through the medullary pyramid. The Bowman's capsule encloses the glomerulus to form Malfingia or renal corpuscle. Urine formation involves three main processes, that is filtration, reabsorption and secretion. Filtration is a non-selective process performed by the glomerulus using the glomerular capillary blood pressure. About 1200 ml of blood is filtered by the glomerulus per minute to form 125 ml of filtrate in Bowman's capsule per minute. JGA, a specialized portion of the nephrons, plays a significant role in the regulation of GFR. Nearly 99% of reabsorption of the filtrate takes place through different parts of the nephron. PCT is the major site of reabsorption and selective secretion. Henle's loop primarily helps to maintain osmolar gradient 300 m/osmol per liter to 1200 milliosmol per liter within the kidney interstitium. DCT and collecting duct allow extensive reabsorption of water and certain electrolytes, which helps in osmoregulation. H+, K+, and ammonia could be secreted into the filtrate by the tubules to maintain the ionic balance and the pH of the body fluids. A countercurrent mechanism operates between the two limbs of the Henle's loop and those of fossa recta, capillary parallel to Henle's loop. The filtrate gets concentrated as it moves down the descending limb, but it is diluted by the ascending limb. Electrolyte and urea are retained in the interstitium by this arrangement. DCT and collecting duct concentrate the filtrate about 4 times, that is from 300 milliosmol per liter to 1200 milliosmol per liter, an excellent mechanism of conservation of water. Urine is stored in the urinary bladder till a voluntary signal from CNS carries out its release through the urethra, that is maturation. Skin, lungs and liver also assist in excretion. In this way, we completed the reading and discussing important points regarding Neet exam. If you liked the video, then don't forget to subscribe to the channel Medical Duo. Thank you.